Okay, good afternoon, everybody. I'm Leo Mori, and I will be as quick as possible because I don't have so much time, I think. So I will introduce you BioimageIT, our middleware of data management and data analysis. And it has been developed by the STED Serpico team. So to talk about BioimageIT, I will answer to a few questions. So the first one is when to use BioimageIT. So to have some context, you have here a very schematic view of a scientific study. And I will not describe all the steps because I don't have the time. But I will focus on the part after the measurements and the acquisition of the raw data. And so BioimageIT manages the raw data and the metadata. It processes the raw data and optionally with pipelines. I will talk about it later. It saves the program, the algorithm, and pipeline parameters, and makes easy to reproduce the experiment by respecting the fair principle. I won't explain it for you because Fabrice Cordelier just did it. Why should we use BioimageIT? Because there is here an example of a pipeline. So you have your raw data, you stored it maybe locally or on a remote database, and you want to do some dequeuing or some deconvolution or tracking, and then you want to visualize your processed data. But you have to do it manually, so manual construction of the pipelines. You have to manually manage your data, and there is no automatic recording of the parameters, and and the pipelines are only described in Lab Notebook. So with BioimageIT, you have all of this in an unique interface, and you can use the functionalities without any coding skills. You don't have to re-implement existing tools. There are already some tools on the BioimageIT, but if you want to add your own tools, it's fairly easy to do it. The idea is also to design your own pipelines without any coding. BioimageIT manage automatically the data and the metadata, and also record automatically the parameters. To do a parallel with this example, you can do all of this within the BioimageIT interface. You don't have to jump between several softwares. Next question is how does it work, or how is it made? So I will explain it with 6 pillars. So the first one is the pillar of the format. So BioimageIT can read, convert, or generate different types of formats. It could be TIFF, it could be ZAR, which is compressed type of images. You can also generate CSV if you have analyze some things on your images. If you want to read proprietary formats, we can use bioformats. The second one is the tool wrapping. So every tool integrated in BioimageIT comes with his own wrapper. And what is a wrapper is a description file, which makes the bond between the algorithm, your tool, and the user interface. So the third one is the runners, which it's in the name, they run the tool from the wrapper. The data management part, yes, the data sets you import in BioimageIT are represented in experimentation as experimentation. So I said it auto-saves the data and the metadata, and it can communicate with external databases such as Homero or CID. Yes, about the visualization. So the visualization is integrated also in the graphic interface. So we can use Napari or Matplotlib or Qtable depending on your data. And the last one is the data interaction with the reading of the annotations. So this is a glimpse of the interface when you import your data in BioimageIT. So the data is stored by experiment, and the data annotations are read and extracted from the file's metadata. And there are multiple storage backends, local, remote, and Homero, for instance. On the side of data analysis, this is the interface when you open a tool in BioimageIT. So you can run the data processing tools from existing softwares. You can explore the parameters. As you can see, there are buttons and sliders. And you can also run on your full data set without any coding. So these are some examples of tools. So this is a poster sent by Gabriel Le Goff from the Imagic facility in Institut Cochin. And we focused on the part of the segmentation and extraction. And as you can see, we integrated in BioimageIT cell pose, which can work on GPU. Also in segmentation, we have another example, such as StarDist. The idea is to show you that we are not limiting on the programming language. You can import, add some Python script or C++ or Java or Fiji macro. You are really free on that. To finish, some recent achievements in perspectives. In June, we participated to the Century Hackathon in Marseille, where we presented BioimageIT, and in that time, we also could add some new functionalities. J'ai bientôt fini. And 2 months ago, the article of BioimageIT has been published in Nature Methods. We are pretty proud about that. And for the perspectives, so my job is to deploy BioimageIT on several facilities of the France-BioImaging infrastructures. We are also always open to add some new tools and to show and train people to add new tools by themselves. We're also working on the pipeline designer. Which is typically boxes that you drag and drop without coding. Next, some information about where BioimageIT has been presented or installed, and where I will go, who I will contact for future installation. So thank you for your attention. And if you have questions, I will answer later, not now.
So this, yeah,
il fonctionne.
The second project presented will be the CloudFish project, and Xavier DeVos from Marcelo Nolman team is going to present. Xavier.
Perfect. Okay. I'm Xavier DeVos from the CBS at Montpellier. I work with Marcelo Nolman, Brian Muller, and Thomas Walter on the CloudFish project. It's a development of a user-friendly tool for the analysis of single molecule RNA and DNA fish images. So many biological processes require to detect transcription or chromatin structure in single cells. Typically, this requires measurements of multiple DNA or RNA species at once. But the classical microscopes are limited by the number of different colors identifiable on the same image due to the overlap of emission spectra, like you can see here. You can't make on the same image hundreds of different colors. So recent solution is to couple fluorescent microscopes with microfluidics device to sequentially image multiple DNA or RNA fluorescent probes. To do this, we take one probe by one, and the sequentially cycle will injected the probes, imaged, photobleached, and pass to the other, and this tens of times. But this process generates hundreds or thousands of 3D images, and just for one experiment. So developing of automatic and robust analysis pipelines are really critical to analyze this data. In the FBI network, to analyze pipeline already exist. There is FishQuant is for the RNA fish analysis. It's FishQuant segment cells detect the RNA localization and allow to analyze the spatial distribution pattern. So you can localize here, that is concentration. And the second is PolarIMAGING, is for the multiplex DNA fish data. PolarIMAGING will register 3D images, segment, and extract also the detect the DNA fish signal to build after the chromatin interaction matrix to understand the structure of chromatin. So in the CloudFish project, there is two main goals. First is to unify the two tools into a common and generic Python library of feature. And this library is for the all fish data analysis. And we want to make this library improvable by the community. And the second goal is to develop typical usage pipeline to use this library. And we integrate the pipelines inside a user-friendly graphical interface to easily manipulate the parameters and to visualize results of a run. So to start, we have to build HappyFish, a common function library. It's a toolbox made to be accessible for everyone who want to make data fish analysis. Actually, HappyFish is the merge of function from BigFish and from PolarIMAGING. BigFish is also FishQuant, to be quick. And HappyFish is a community-driven and collaborative project who welcomes collaboration from external developers. But the issue is that all users are not comfortable with Python script development. So the second step is to implement a graphical interface in a user-friendly way. To start, we are initially developing an API plugin to use HappyFish. This plugin will allow the user to manipulate, configure the parameters, test if it's the good configuration, And after, you can execute your pipelines, and with NAPARI plugin, explore the results. In the future, we are open for other possibility we're exploring other graphical interface like ImJoy or BioimageIT, like you have seen before. And this meeting is a great opportunity to also discuss about this. And to conclude, during my first year of contract, I start to make a big refactoring of the PyM software to make modular architecture. I rewrote the documentation and put it online with read the docs. I work on tutorials to facilitate usage of PyM for the user. And also I set up non-regression tests to ensure a smooth transition from PyM to HappyFish. And also I make a friendly installation of the software based on pip. Thanks for your attention. Maybe some question.
But we can move on.
Okay. Ciao.
Next speaker is Abdelghani Nous from Jean-Baptiste Sibarita team. And he will present the tech transfer for the BIT HCS SMLM project.
So hello everyone, I'm Abdel Nous, as said, and today I will be presenting you the technology transfer of a single molecule-based high content screening platform to the Bordeaux Imaging Center. So to give a little bit more of detail of this project, so there is 3 teams involved to the transfer. The first one is the quantitative imaging of the cell team of Jean-Baptiste Sibarita. The second one is the dynamic of synapse organization and function with Mathieu Salos as its reference. And the third structure is the Bordeaux Imaging Center with Magali Mondin as referent. And the team of Jean-Baptiste Sibarita has developed a single molecule-based high content screening platform that it is used to image fix or living cell in a single molecule context. And we are using multi-well plate devices to do that. And this type of experiment generates a lot of data that can be analyzed in a quantitative manner. And the goal of this is to decipher some fluorophore photophysics, molecular dynamic behavior, and so on. And the goal of this project is to transfer these whole things to the Bordeaux Imaging Center. So more detail about the platform. So the platform is composed of 2 blocks, if we can say this like this. So there is the microscope linked to the camera used for the acquisition, and the camera is interfaced with Metamorph, which is the acquisition software. The Metamorph contains a plugin called Wave Tracer, and this plugin is used during the acquisition to do live detection of the molecules, live super resolution reconstruction, but also live statistics and so on. And the other block of this platform is another plugin of Metamorph, which is the high content screening acquisition plugin. And this plugin is used to set up the high content screening experiment, such as, for example, selecting the well we want, as you can see. And if we look at the global acquisition and analysis workflow, so we have our microscope connected to the camera that acquires some data. These data are analyzed during the acquisition. So we are localizing the molecule and the traffic if we are doing some trajectory experiment. And after that, there is an offline analysis that can be done to extract more photophysical properties or other characteristics of the proteins. And after that, with some homemade or already existing software, we are able to do metadata extraction, analysis, statistical visualization, and so on. So this platform has been described and published in 2017 in this paper. So here we have an example of, it's actually a 3D distorm imaging of a wall multiplate. And it took approximately 8 hours to do the molecule detection and the reconstruction steps. So now a little bit of timeline for this project. So it started in October 2021, and the first step was to transfer the plugin to the Bordeaux Imaging Center and to train and test this plugin to make it accessible and usable for the user of the BIC. And this step is done, so the plugin is actually accessible to the user of the platform, since the BIC already have the microscope and the setup required. The second step is to install a flat top illumination to upgrade the illumination system and to create a sort of even illumination over the whole samples. After that, there is two big parts that are quite of superimposed. So the first one is the development of a GUI inside Metamorph or not to handle this quantitative data in a user-friendly manner. And the second big part is the pilot. So there is this pilot, this really big first project with the platform inside the BIC to screen fluorescent protein, but also synaptic protein. And this is called the Stable FP project. So more detail about this project. So it is founded by the Agence nationale de recherche. And the goal of this project is to increase the number of fluorescent protein available by adding new photoconvertible fluorescent protein inside this already existing toolbox. And this protein need to have some increase or better characteristic, such as, for example, a near complete green to red photoconversion, an increased photon budget, and so on, but they need to preserve their main properties. So for that, it is a project, it's a collaboration between Jean-Baptiste Sibarita's team, Mathieu Saint-Los's team, and the BIC in Bordeaux, but also with Dominique Bourgeois from the IBS in Grenoble. And the goal is to do a quantitative characterization and study of several fluorescent proteins. So here is a typical example of an experiment we could set up, for example. So here we are testing several concentration with several fluorophore, but we could also test different illumination scheme, different buffer, different acquisition condition, and so on. And the advantage of this platform is that the comparison and the analysis will be unbiased, since the technique, the HS, bring the standardization. So all the wells are acquired in the same manner. And this will be used to benchmark media, dyes, or probes by using the photophysical metrics we have. So now we'll talk about the advance of this project. I will be mainly focusing on the two big parts in blue and green. So for that, here is a beautiful single molecule image of MEOS 3.2 protein. And so for that, the first step during the analysis, after acquiring the image, we want to detect each single molecule which are blinking. And at the end, for each molecule, we have its localization, its intensity, its coordinate, and so on. And it's the same thing that is done during the acquisition with the other plugin, but we can do it also after the acquisition. And with this output, we are able, with another software, to put in the same plane all the molecule detected. And after a clustering step, we are able to, for each cluster, to have information about the lifetime, the number of blink, etc., inside a cluster. And so to link this to the transfer to the BIC, the idea is to have this kind of information and this kind of extraction in a GUI inside Metamorph. And for the Stable FP project, it is typically the kind of photophysical properties we are interested in. So this is the link between these two steps. Well, it's a conclusion. So to sum up, the first step of transferring the plugin is done. There is still this flash stop illumination that need to be added to the setup. Regarding the GUI, there are already some tools developed and available, but they need to be added as a GUI inside Metamorph. And for the screening and the stable FP project, so the sample preparation and the imaging condition have been validated, and now the future is in the future we have to screen other protein, and that's it.
Thank you, Abdel. We have to move on, and Tao Laurent is going to present the Morphonet project from the MNL4 team.
Okay, can everybody hear me? Okay, so thank you for being here. I'm Tao Laurent. I'm an engineer in LIRM at Montpellier, and I'm working on Morphonet, and more exactly the tech transfer aspect of this project. Just to give a quick bit of context, and I'm going to be very fast. So I'm working on the Morphonet project, which is an interactive 3D+T web-based data browser on which you can visualize meshes and images. And so you can see a little video of an Ascidian embryo. And that's all I'm going to say. If you want to know more, you can go on morphonet.org. But this is the main project, and it also has a couple of other tools like a Python API and a data curation tool that at the moment requires you to use the Python API. So as you can see, it's a pretty interdisciplinary project because the target audience is biologists, but of course it is based on a lot of concepts of computer science, like computer graphics and so on. So in this context, the main idea was to make the tools easily available for biologists. So this goes into three kind of axes. The first one being the interoperability between Morphonet and the tools of the community, such as Fiji, Homero, Napari, Maris, or many others. Also, it is with the formation and user help for the users that are interested in Morphonet in FBI nodes, and also into an ongoing effort to have some overall improvements to reduce the need for computer science expertise, which is often required by biologists to use tools. So I'm just going to go quickly through a bunch of contributions that have been made in this regard. So the first one was to develop what we call Morphonet J plugin, which is a Fiji plugin that we did with the idea of going from your segmented image in Fiji to your data set that you can view on Morphonet in just one click. So you have your segmented images, which are converted into meshes in one click, and they're either directly uploaded if you have an account, or they're just converted locally if you do not. You can also upload directly your images and upload additional data to your datasets using Fiji tables. And so I'm just quickly going to show it. So here is an image of Drosophila embryo. And by opening the plugin, by just logging on and so detecting your image, you can, in a couple of configuration tweaking and just one click, convert your image into a mesh, which is directly going to be uploaded onto Morphonet. And then you could use the whole range of tools that are available to you to just view, analyze, and augment your dataset, as you can see here. So this also paved the way for other plugins such as in Napari, but I'll talk about this a bit later. On two other things that we did was to make the Morphonet more available and easier to use. So for instance, one of the other tasks I did was to rework the annotation menu. Using this menu, you can give labels to cells and also colors, shaders. And the idea was to rework it so biologist users could use shaders and have a bunch of features without any knowledge in computer graphics. And also I did something such as speeding up the loading and visualization of raw images using a server-side conversion, then again to be able to have a smoother, faster experience and make the Morphonet more available. Also, I worked on a channel system for meshes and raw images, so you can view more data and have more complete data sets. For instance, you could have meshes of membranes of cells and nuclei, and like different red, green, blue channels for your raw images. And obviously, I work on overall maintenance and development of new features, with the main point being that we try to steer development with the biologist users. It is their need for new features that drives development. And so the idea of tech transfer was also to do presentation for interested users in FBI nodes, to present the tools, the features, to do some demos with our data or their data, and to give personalized help, because understanding what the different challenges are in other teams allows us to manage different data types and to respond to the needs of the community. And also it allows you to have discussion about improvements, new features, and so on. And so we did this in Montpellier, in MRI, in Rennes, and we wanted to do this in Nantes, but we couldn't really, so maybe we could do this again and perhaps do other nodes, hopefully. And so finally, for a bit for perspectives, there's still a lot to do, whether it is for interoperability with new plugins such as Napari, for instance, and we still have a lot of features to do for ease of use and accessibility. And so in this regard, the next new feature that we want to do is the complete standalone, which should allow any user to have great performances and no need for either an installation or knowledge of coding, such as using Python. And also it would have VR and more. And so we would like to continue also the interventions in FBI node as part of the tech transfer to better understand what the community needs. And so I'd like to thank everybody that worked with us on this, and thank you for your attention.
Thank you, Tao. Now we switch to the outreach call presentation, and César Gustavo Valadez is going to present the PolarIMAGing project from Sophie Bracelet in France.
Hello, everybody. It will be very short because I only have 2 minutes. So this is was one of the projects that was successful from the call of outreaching. So the team leader is Sophie Bracelet from Institut Fresnel. In fact, she couldn't stay here, but I'm presenting. I'm from the Serpico State team. So one of the user teams. So as I mentioned, there is the leader team. There is two developers teams. That the first one is from Sophie Bracelet, Institut Fresnel. The second one is from Bas and Hash from Institut Curie. Then there are 3 teams more focused in biological imaging, in biology. And one is the team of Serpico State. We are more focused in image processing. Then there are 2 external teams, one from Germany and the second one from Strasbourg University. So the main goal of this project is to combine one technique that was developed previously by Institut Fresnel that is called for PolarIMAGing. So with another technique that was developed already many years in the Salamero teams, currently Serpico State team, that is multi-angle turf. So the idea is to combine this technique of polarization that allows to have orientation with the 3D information from the multi-angle turf. Currently, this project uses STAR. In fact, we start to do the tech transfer between the postdoc Miguel Sison from Institut Fresnel and the research engineer Ludovic Lecomte at Serpico State team. So we already mounted the 2 main parts. So now we are working to optimize the polarization part. As I mentioned, the goal is to combine this possibility to have orientation and the 3D information, as I show here, and also the organization in the membrane. So this project has different funding. In fact, it started as a groundbreaking project from LabEx, and now with the outreaching and tech transfer with the France-BioImaging, but it's also included for the future and INER that was just getting by the team that is more focused in image analysis, and finally other projects as microfabrication. Okay, thank you.
Thank you, César. And now the last but not least, the virtual microscopy project by Emmanuel Faure.
Hello again. So I already thanks everyone. That's easier to do it because I have 3 minutes. We told me 3 minutes, not 2. So this project was actually initiated with a Naviscope project at the NRIA. So thank you, Charles, for that. It was a very, very starting point. We did, Gwendal just finished his PhD working on how can we visualize let's say images from different microscope, okay, and interact with them and etc. So it was very, very interesting PhD, supervised also by Ferran and Charles. Kevin, after, which is an engineer in this project, is working now since maybe a year, try to how can we now transfer all this PhD work inside something more usable for biologists and etc. And this is a team that is involved also. So what do we want to do? I have 2 slides. What do we want to do? It's first be able to load the data from, let's say, any type of microscope. Okay, well, focusing on 3D data. Okay, because with virtual reality, you don't want to play with 2D. That's not the purpose. It's to have another type of interactions with this, using all this environment, but for 3D or 3D plus T dataset or with channels and etc. Okay. So first things that we will do, the phase 1, is to be able to load all the what we call the raw data from direct resolution microscope, not necessarily the segmented data. Okay, this is an example of the video that now we can load. This is a light check microscopy images in 3D, and so here it's in 3D plus T. You can play and etc. But we need to improve that a lot because that was really developed for our type of data. That's the thing that we're working on. And so for that, the main idea is that from a user, you just directly go upload your data on the browsers and be able to load them with virtual reality headset. Okay, not necessarily upload the data. You will be able to just load it on your computer, okay, without uploading anything on our server, and you will be directly be able to visualize your just acquired data. Okay, maybe it will take a couple of minutes, but that's the idea. So we have some already videos that we are work in progress. Okay, so this is, for example, all the time that you can see in a spiral around you, all the things that you can cut with your hand and see what happened inside. So this is not on raw data. This is on segmented data. Okay, wait, the project starts in January actually. Okay, so this is here the times around you and etc. So if you're interested, we can discuss that later. And because it's the last slide that I will show you, I have an announcement for the next years of the annual meeting of FBI. It will look like that. So thank you very much.
So for the sake of time, my introduction will be very brief. Our next speaker is Antje Kepler. She's the director of the BioHub of Euro-BioImaging. And she will talk to us about user access and funding opportunities through Euro-BioImaging, 2 very important points, because as you know, one of our mission as infrastructures is to facilitate the access of the scientists to our state-of-the-art instrument we have on our facilities. So Antje.
Yeah, thanks a lot, Caroline. And it's really a pleasure being here. As this is the last talk between us and the museum, I will be very concise and much shorter than the 50 minutes indicated. Also, John Eriksson, the Director General of Euro-BioImaging, will give another presentation on Euro-BioImaging again, so we can discuss details tomorrow. So Euro-BioImaging, as all of you hopefully know, and I'm preaching here to the choir, is a distributed European infrastructure with 17 countries and 1 international organization supporting it. And together we at the moment have 35 nodes, of which France-BioImaging is 1. And together they make up 173 individual imaging platforms today. Together they offer more than 90 different technologies, and I'm not going into any detail here, but it ranges really from individual molecule imaging up to imaging of humans and populations. Researchers can reach us. Any researcher on this planet can reach the Euro-BioImaging services through the Euro-BioImaging website, where we also have the Euro-BioImaging web access portal, where the technologies, nodes, and also the data services can be found. then the researcher usually would reach out to you first as the experts at the nodes to understand what is possible, what is feasible. And then if they do not know yet to which node they want to go, they reach out to the team at the Euro-BioImaging hub to ask us for advice. After this initial consultation, they submit online their application. They might get scientific advice. If their project has already been previously evaluated for funding, they can also take the fast track, and the application will be forwarded directly to the node to you for technical feasibility check, and also if you have time and capacity for the incoming user. And then the service provision can start. So this is probably the service provision procedure, as you also know it from France-BioImaging. Euro-BioImaging is open to all scientists, no matter where they're from, no matter at which level they are in their career, if they're from academia or industry. And when it comes to the question, who of my users can I send through Euro-BioImaging, you can send all users that are external to your department. So any user that is not legally affiliated with the same institute can apply to your facility through Euro-BioImaging. And with this, I'm already coming to the funding part. As I promised, I would try to be fast. So on the Euro-BioImaging website, let's assume you have a user coming in and you will charge them a fee and they do not know how to cover that fee. You can send them to us. We will first check with the user on the website if there's maybe a call open that might fit the user's purpose and background. And also, we often now recommend to the incoming scientists to check when they apply for the next grant. Most funders in Europe and also in the European Union, they are now accepting when the user upfront puts the request for funding for user access into their proposal, and they should really do this. So if you're aware of scientists applying for their next grant, tell them, you can benefit my facility if you already put in those costs. Exactly a year ago, Euro-BioImaging opened a pilot user access fund where we put from the COVID-19 savings 50,000 euros out there and said, we invite all scientists to apply for access. We wanted to test with this how large would be the request, can we do this, and also then provide that data to the member states that are funding Euro-BioImaging ERIC. So we put out the 50,000 per user. We said 5,000, that's the upper limit. And we expected 15, 20 applications. We got 85. That was just a few days before Christmas. But the team was so excited about this good news, we didn't mind working a night shift just ahead of Christmas Eve. And we worked then through the process. And these 85 applicants presented 32 different countries, also quite a few international requests coming in from overseas. And 75% of these applications were transnational access, so asking for access going from one country to another in Europe. With 50,000 euros, we could fund 10 scientific impactful projects. It was really, really hard for the reviewers to pick those among all the excellent projects that came in. And then we want to say thank you again, merci beaucoup, for helping us out at this point, because France-BioImaging got quite a few strong proposals at that time. We could not have funded all of them, but you were able to take 9 in addition to the 10 and give them France-BioImaging access with France-BioImaging funding. So thank you very much. Those users are very grateful, and I bring you here one example. So Valentin Boulan says, working with the lab in Nantes was a very nice experience for me. I learned a new technique. There's a potential to work more together again. So he was here to use double illumination light sheet microscopy to get a 3D view of myofauna. Another user coming to France-BioImaging who was funded through the Euro-BioImaging funding was Andrew Boyce. He came from Canada and he visited the BIC, the Bordeaux Imaging Center, and he says, sushi is a very exciting technique for studying brain tissue, but sample preparation is tricky. Coming to the Bordeaux Imaging Center to work with a person who developed this approach and get expert training is a dream come true and will make a big difference to my research. And these are the quotes that you should be using when promoting France-BioImaging with a national roadmap, but also in Bordeaux. So going from the pilot user access fund last year to the EU landscape. So the Euro-BioImaging Hub team has been quite busy and also successful in applying for EU funding for Euro-BioImaging. And this is funding that goes to the users of Euro-BioImaging as well as underpinning opportunities for the nodes. And today I only have time to present 2 of these manifold projects to you. So we have projects now funded in Euro-BioImaging, 3 for transnational access, that's AgroSurf, EasyDoor, and CanSurf, as well as AirRemote to underpin remote access provision to training and instruments. Then we have quite a few projects in the domain of fair data and services, and I've seen so many exciting presentations here earlier today. Please reach out to us, join the expert group on image data management and analysis, and learn more about these opportunities, because these projects are for you as well. And you can also benefit from participating in the hackathons, but also applying in the open calls. And then we have the new technology developments, the ANERES project, where France-BioImaging is a partner, and Imagine and Impress, where we have calls for the nodes to come in the next years. But now to EasyDoor. So EasyDoor is a funded project for transnational user access in the domain of infectious diseases. It's up and running. We have 9 of the 35 nodes in Euro-BioImaging are participating, including France-BioImaging. And you can receive funding for your user projects when they are working on SARS-CoV-2, risk group 4 pathogens, respiratory pathogens, vector-borne pathogens, or any other pathogen. Reach out to us. We can help you if your user project is eligible, and please spread the word. So the funding is there now, and the sooner we use it up in Euro-BioImaging, the more we can get from the project. The other 2 projects, CanSurf and AgroSurf, are a bit younger, so they just started officially. They are now in the process of defining the procedures and calls and access management, which is always a lot of fun for the coordinators as well as us. But CanSurf will be the next one to watch out for. And if you know already users in cancer research coming to your facilities, soon there will be funding to fund for their access, travel, and accommodation. My last content slide, AI for Life. That's a project that is coordinated scientifically by the Human Technopole and EMBL, and administratively by Euro-BioImaging ERIC. And here we will have opportunities for nodes. Again, open calls for training, meetings, hackathons, and yeah, watch out for this. We are always in touch with Caroline about this. And thank you very much. Merci beaucoup. And I wish us all a nice evening.
Thanks a lot. So we have time for some very short questions, if any. Yes.
Yes, no, I just want to make a comment. That's to remind you that we provide money for the people that access your facilities, as long as they're not from your perimeter, if they go through the UBI portal. So please use that.
Okay, so now we will have to go, like in 5 minutes, we will be leaving from here for the social event. So we work together. It's Les Machines de Lille, if you're looking on your phone. One thing also is that tomorrow there will be the working group meeting. And this will be displayed in the corridor everywhere. But basically, if you didn't register to any working group meetings, just check with me at the reception, but otherwise you will go to the amphitheater, okay? And for other people, there will be some rooms that will be displayed also tomorrow. So it starts at 8:45, okay? So that will be displayed everywhere. And there will be some people to bring you to some room when it's upstairs. Just there were 3 people that were registered to this working group. And unfortunately, it is cancelled because the animators are not here. So these people can feel free to go to any other group. And that's it. So thank you for this first day of operating France-BioImaging for 10 years. And now tomorrow will be the scientific advances of 10 years also. Thank you very much. Let some stuff. Pour les gens qui ont laissé des choses dans la pièce 1, elle est ouverte, hein, vous pouvez y aller.